traditionally eat eel to
but that's
eat.
about it. You really don't get expression in other areas. So this is really nice for doing math.
Residents were asked if they
What
eat
we're
eel.
going to do here is we're going to use the distribution
he's
of
gone.
morphology to actually
He says
study
he's
where
never
the
eaten eel.
power is spreading just to its projection areas. And in this model, the projection areas here, you can see the projection areas
It's quite true that he's never eaten eel. So why don't they eat eel in this area? The chief priest at an local Shinto shrine had this to say.
Middle layer of the molecular layer of the dentate gyrus, the projection area from the endocrine cortex. And you see how
Nishikoto says they never eat eel because they believe the fish is a servant of God.
What we're really interested in is
A sign near a river says people protect eels as servants of the gods and never eat them.
Cells. And from there, does it cross from other areas of the hippocampus to areas that start to be affected later in the disease? And if they start to spread up here, these areas are not related unless there's to a synapse to areas down here. And this is then transsynaptic spread. So we're using this sort of approach.
People in the area have respected eels since that time.
Pathology developing. Now we're not all about pathology. We've learned our lessons that perhaps pathological lesions aren't actually the disease. Maybe the tangle pathology or amyloid plaque is not the disease. Maybe you're looking at an intermediate, a beta form or a tau form that you don't see in a tangle. Also, maybe the disease is not all about the control or even intermediates. Maybe it's actually just cellular dysfunction. Something
Nishikoto says eels help people to clean rice pots.
Other ways, and that the pathology is just one aspect of this. So we're coupling these mapping studies with studies using fMRI.
So, does he eat eel himself?
Of regions, the
He says no, never. He says he would never have even a single bite or touch one with chopsticks. He says people tell him he's straight-laced, but that's fine. But people in other areas eat eel to get energy. This person says he feels sorry to see eels being grilled.
Data, but I won't show you. It's really too preliminary to bring it up. But we can see dysfunction down here by fMRI. We have not
severe heat will continue around the country tomorrow. Daytime highs will be 38 degrees in Kumagaya and 37 in Laibashi and Kofu. The Meteorological Agency has issued an alert for hot temperatures and advised people to take precautions against heat stroke.
Different epitopes of our molecules of interest and see if the pattern distribution of them is the same or different, and what it is about those different epitopes that perhaps is giving us a different pattern. So here again, this is the endocrine cortex where this is expressed. And we see this is an antibody called MP1, the confirmation abnormal form of tau. It's a human-specific, is the control showing no staining. It's an abnormal confirmation. You can see it's in the projection area. So this is what tau just slows down the
temperatures expected to climb. Be sure to take lots of fluid and turn on the air conditioner to prevent heat stroke.
This is CP13, which is screening to a 5. This has
There are exactly 5 years before the start of the Olympic Games. Japan has set a goal of getting the 5th highest number of gold medals in the 2020 Games. To achieve the goal, special programs have already started to train talented young athletes.
We also think, but we haven't had old enough minds really.
The organising committee unveiled the official logos for the Olympics and Paralympics today.
And this sort of pattern would be very, very nice to show. If it's found in older mouse and it gets worse, this would be exactly what we'd expect if you've got some kind of spread of pathology. This would be very exciting if this was human.
Today marks
because then that would tell you that it's spread of a molecule transsynaptically. Unfortunately, we don't have phospho antibodies to recognize human tau. So this recognizes both mouse and human. And this is the control mouse. You can see that some of the mouse tau is recognized by the antibody. So we need a lot more antibodies to be able to do this sort of study and start to map together what's going on the distribution of tauopathy and tau forms. Just as a comparison, we've done exactly the same thing with an invisible ACC transgene, which cross back to our neuropathy detector, and you can immediately see there's a real difference between how tau is distributed both in the cells and how it flows down into the projection areas, and how ATP and A beta is distributed and where it goes to. This is a human antibody, and unfortunately this is meant to be A beta specific, but I don't think it is. This is ATP post-beta. Here you'll see actually we've actually aged these a little longer, and they've actually got amyloid plaques forming down there in the endocrine cortex. So this is a pretty robust model with amyloid plaques forming there. Again, we'll be using these to contrast our two different pathological regions. We can cross them together, we can turn off the pathologies, by use of tetracycline, etc. And we can actually see, we've already started to see some differences in distribution that might suggest correlates in the human AD brain. One thing of particular note is the sparing of the dentate gyrus in this APP model. You usually don't see that spared in other mouse models, suggesting that this layer is actually very much affected by what goes on in the entorhinal cortex. And this is very similar to what you see in human AD. You see real sparing of the dentate gyrus in human AD and not usually. So I think this is actually very interesting and telling us that the entorhinal cortex perhaps is really a very fundamentally affected area in Alzheimer's disease. So I wanted to talk a little bit now about the mechanisms of tauopathy and how we're trying to intervene therapeutically. And this is a slide showing all of the different mechanisms that we can envisage going on. We don't know what initiates tauopathy other than when you've got mutations in tau, but we think it's probably some age-related degeneration of several systems. We have seen that elevated A beta can provoke and accelerate tauopathy. If you cross an APP mouse and a tau mouse, you get evoked pathology in those mice. And we're not sure that there's something wrong with the tau aggregate, or if it's a loss of tau's normal function. And tau's normal function, as I said, is to stabilize microtubules. So you could have a significant disease effect if you lose its normal function, even if the tangles themselves are not important. It's something about loss of normal function. So here we have the hyperphosphorylated tau that forms because tau forms off the microtubules. It becomes a target for kinases and for phosphatases, and it can become hyperphosphorylated, accumulating into these aggregates that we see in the brain. This is some of the pathological events. Now, these are double-sided arrows. We don't know which one comes first. We don't know which is related to which.
Current interest, and that is stem cell research. And particularly, George Daly, our speaker, is going to present to us some new and exciting data, some of which you might have seen a little buzz about in the last few days, about some new information about induced pluripotent stem cells, and I think you're in for a really interesting and informative scientific seminar. George is the Samuel E. Lux IV Professor of Hematology. He's also the director of the Stem Cell Transplantation Program at Children's Hospital, and he is professor of biological chemistry and molecular pharmacology and pediatrics at Harvard Medical School. He has obtained the MD and the PhD from Harvard and MIT, respectively. And then after internship in medicine and postdoctoral fellowship, all of those in Boston, has been a very productive faculty member since that time, including an appointment as an investigator in the Howard Hughes Medical Institute as of 2008. He's been elected to the ASCI and the AAP, and we at NIH are particularly proud of being able to point to the fact that he was one of the first Pioneer Awards achievements, and that of some of those other Pioneer awardees as an indication of what a successful program that has turned out to be. He is not only someone who has led